So after receiving his PhD at the Institute of Biophysics in Chinese Academy of Science and completing his postdoctorate at the University of Chicago, he was promoted assistant professor in Chicago. And since 2016, he joined Shanghai Jiao Tong University School of Medicine as a professor and a PI, and to understand new mechanism of tumor immune escape in the context of innate immune sensing and T-cell stemness reprogramming. And today, he will share his data about cytosolic DNA sensing. So thank you, Dr. Deng, to be with us, and thank you for your talk.
Thanks for your introduction. Good morning to friends, and good morning to Chinese friends. I'm Liufu at Shanghai University School of Medicine. Today, I'm very glad to share my story to all of you. Before introduce my research, I want to have a brief introduction of my research background. There's one concept in the tumor immunity. It's a cancer immunoediting, which was proposed by Dr. Robert Schreiber. So I think cancer immunoediting is an extrinsic tumor suppressor mechanism, and it consists of three stages. The first stage is elimination, and another is equilibrium. The last one is escape. In this stage, both innate and adaptive immune cells work together to kill tumor cells. If tumor cells is not destroyed in this stage, and in this stage, both T cell and cytokine like interferon gamma and IL-12 are required to maintain tumor dormancy. If this balance is broken, then the tumor cell will go to escape stage. So there's multiple mechanisms for tumor escape, like the immune suppressive immune cells and immune suppressive factors like TGF-beta, L-gene, and IDO. So there's a cancer immune cycle in tumor microenvironment. So you ought to generate effective anti-tumor adaptive immune response. So each step should work together and to get a robust adaptive immune response. So like in this period, innate immune sensing is the basis for the whole immune response. If innate immune sensing doesn't work very well, so the T cell response will get impaired. So usually in cancer patients, the cancer immune cycle does not perform very well because of tumor immune escape. So thus, the central goal of immunotherapy is to push forward the cancer immune cycle. So here's my research interest. We look at the cytosolic DNA sensing in the tumor microenvironment. You know, there are a lot of immune cells like innate immune cell, dendritic cell, and the killer cell like CD8 T cell and natural killer cells. So we are interested in how the innate immune sensing is initiated and how tumor cell get escape of innate immune sensing. And we want to know the new function of innate immune sensing in tumor microenvironment. So next, I want to share two parts with you about our research. The first, I want to share with you the cerebral CGAS pathway in T cell therapy. So as you know, CD8 T cell is very essential in cancer therapy. It not only in immunotherapy like T cell therapy and immune checkpoint blockade, it can also be needed in conventional therapy like radiotherapy and chemotherapy, also in target therapy. So that's one T cell status in tumor microenvironment. We call it a T cell exhaustion because it's due to because it's a chronic stimulation. So the T cell get exhausted. Compared to the memory T cell, exhausted T cell have a decreased effect function and proliferative capacity, partially because the overexpression of PD-1. Actually, there are two subsets of exhausted T cell. One we call is early exhaustion T cell. Another is a terminal exhausted T cell. So they express different level of immune suppressive inhibitor like PD-1. So in this subpopulation, they have a mild expression. In this, they have a higher expression. Also in this population, TIM3 is highly expressed. Another is they have a different transcription factor and stem like marker like TCF1 in this population is positive, and SLAM M6 is positive. In this population, they are negative. Also, exhausted T cell have a TOX1 positive. So recent study have already shown why immune checkpoint blockade works well, or T cell therapy works well, because in this therapy they can rescue early exhaustion T cell, not terminal exhaustion T cell, because for terminal exhaustion T cell, there's no space to rescue them. So here our question is, everybody have already know the role of cytosolic gene sensing, especially CGAS gene pathway works in the innate immune cell and tumor cell, but nobody knows the role of CGAS gene pathway in T cells. So to test our hypothesis, we want to we start to examine the expression of CGAS gene pathway in CD T cell from cancer patients. So you can see we use a healthy volunteer as the control. You can see no matter CGAS and STING and TPK1, all of them are decreased in CD T cell from cancer patients with cancer. So this suggests this pathway is totally impaired. So to confirm our expression, our observation, we use a flow cytometric assay. There's one available commercial antibody against the STING. So we get this antibody and to test the STING expression in CD T cell from cancer patients. And you can see STING expression level is very low in cancer patients, not in healthy volunteers. We test 3 types of cancer, like cervical cancer, ovarian cancer, and endometrial cancer. So we can get the consistent conclusion. Definitely, STING is downregulated in cancer patients of CD cell. So it looks like STING is downregulation is a biomarker for patients with cancer. So next, we want to know what's the function of STING in T cells. If STING is downregulated, so what's the effect for T cell therapy? So to test this hypothesis, we utilized one tumor model. So in this model, a tumor cell express 4 antigen SRY, and we use another T cell. It's called T2C mice. All all T cell from these mice can specifically recognize SR1 antigen. So here we can use this model to mimic the T cell therapy in clinic. So first we inject tumor cell into mice subcutaneously. Then when tumor is established, we get naive CD T cell from 2C mice and stimulate them with CD3 and CD28. Also, we add cytokine like L7 and 15, which is the cytokine to maintain T cell stamina. When T cell is activated, then we transfer those activity T cell into mice and see the therapeutic effects of those T cells. So you can see definitely activity 2C cells can control tumor growth very well. If the T cell without CGAS, so the tumor will fail to control. So this suggests intrinsic CGAS in a double transfer T cell is very essential. Is essential for anti-tumor effects. So consistently, if the double transfer T cell is absent of STING, so they also cannot control a tumor very well. So this suggests the intrinsic CGAS STING pathway in infused T cell are very important for T cell therapy. So next, we want to know what's the mechanism why CGAS STING pathway can contribute to T cell therapy. As I mentioned, stem-like T cell is very important for immune blockade and T cell therapy. So next, we want to know where the CGAS STING pathway can regulate the stamina of T cell after transfer. Then we utilized the model I mentioned before in my previous slide. Then we analyzed the T cell in tumor environment. So you can see if the adoptive transfer T cell loss of STING, they will have a decreased TCF1 expression and SLAMF6. So those two are marker for T cell stamina. So this one is a transfer factor. They are intracellular. Another is SLAMF6. They are surface marker for T cell stamina. So this suggests STING in T cells is very essential. It's very important for T cell stamina. So we next want to know where the CGAS plays a similar role. So we transfer the CGAS deficient 2C cell into tumor-bearing mice. Consistently, we can see if the adoptive transfer T cell is absent of CGAS, so the T cell cannot control tumor very well because they have a less TCF1 and SLAMF6 present. So this suggests CGAS pathway contributed to the differentiation of stem cell like CD cells after transfer. That's why T cell can, without CGAS, fail to control tumor. So next, we want to test whether our finding, whether our observation is existed in human T cells. So to test this idea, we get CD T cell from a human patient, and then we isolated naive T cell. Then with the stimulation of CD3, CD28, and cytokines. Also, we add STING agonist into the culture. The STING is C3. So you can see the T cell stamina is significantly increased after the STING agonist addition. So this suggests maybe the pre-treatment of STING agonist before T cell transfer, so we can have a better anti-tumor control. So to test this idea, we utilized an xenograft model, we utilize A549, which is a lung cancer cell line, and which is express CD28, which is the target for T-cell therapy. And we inject this tumor cell into not scared mice. And when tumor is established, we treat the mice with CAR and CAR plus STING agonist. So you can see in solid tumor, the transfer T cell cannot control this lung cancer. I think this observation is consistent to the clinic observation because CAR T cell can work very well in blood tumor, but it cannot work very well in solid tumor. So our observation get the similar results. Very interestingly, if you treated CAR T cell before transfer with STING agonist, so you can have a better tumor control. So this suggests STING priming before a T cell transfer is maybe a good, maybe a promising therapeutic strategy to enhance the current T cell therapy. So this finding also suggests us if we add a STING into the cocktail, so it will have a better anti-tumor control in solid tumors. So here is our work model. So we found intrinsic CGAS pathway is very important for T cell stimulus. The signal transduction is through CGAS can recognize the cytosolic DNA from nuclei of T cell activation. Then this pathway will induce interferon beta. Interferon beta will maintain T cell stimulus by induce T cell expression. So next, I want to share with you the role of CGAS pathway in radiotherapy. So radiotherapy is a broader interest in cancer patients in clinic. About 16 to 70% receive radiotherapy alone or in combination with surgery or chemotherapy. But how radiotherapy works? So the conventional concept, I think, radiation can cause gene damage. This is the mechanism. So the new perspective is that radiation can cause danger signal and antigen, tumor antigen release. So this will activate the dendritic cells, and then dendritic cell will cross prime CD T cells, and then activate T cell will find and target tumor cells. So we think. So we do a series of work in this circle, and we think local radiation can cause a systemic immune response. So in this stage, we call it first response. This we call it a second response. We think a second response is much stronger and longer compared to the first response. So next, I want to share some works of what we did in this circle. So people have already know adaptive immune response is essential for anti-tumor effects of radiation. So how radiation induce adaptive immune response? So it's missed. So because checkpoint interferon is very important in virus control. So first, we want to know whether checkpoint interferon is also contributed to radiation-induced anti-tumor response or anti-radiation-induced anti-tumor effects. When tumor is established, we treat the radiation. At the same time, we locally block the interferon signaling. So you can see radiation can control tumor very well. But if you block the interferon signaling, so the radiation effects will be impaired. So this suggests that type interferon signaling is essential for radiation therapy. So because there's multiple pathway to induce type interferon, which pathway is dominant to mediate type interferon induction? So we test toll-like receptor pathway, but unlikely mediated and TRIP are not involved in releasing induced anti-tumor effects. But very interestingly, if we use the sting optimized, we can see the radiation cannot fail to control tumor. This suggests sting is a major contributor to mediate type interferon induction and to induce anti-tumor effects for radiation. So in this study, we found CGAS sting pathway work as a link between tumor cell damage and adaptive immune response by induced type interferon production. So based on this finding, we asked whether sting pathway is a good is a promising target to improve radiotherapy. So to test this idea, we intratumorally inject SNE agonist after radiation. So you can see this radiation alone treatment. We combine radiation plus SNE agonist. So you can see tumor 70% of tumor are disappeared. So this suggests the radiation can synergistically work with the SNE agonist. So our study also suggests yes, SNE agonist cannot work well. You should combine SNE agonist with other therapy like radiotherapy. So recently, there's one clinical trial released. The clinical trial is get a very negative results because it just uses SNE agonist alone to treat cancer without any selection. So in clinic, we also can see a lot of tumor relapse after radiation treatment. So we want to know what's the reason why there's a tumor relapse after radiation. So to examine our hypothesis or examine the reason why tumor relapse, we look at the immunosuppressive cells in tumor microenvironment after radiation on day 3. So very interestingly, you can see there's one immunosuppressive cell we call it as a monocytic MDSC is a very highly increased in tumor microenvironment on this 3, and this observation is not exist in other innate immune cells like macrophages and granulocytic MDSC. Very interestingly, this increase is dependent on sting. So this suggests sting is can promote the anti-tumor immunity by increase dendritic function and adaptive immune response. But also sting signaling have a dark side, like it can induce monocytic MDSC, which is a immunosuppressive factor. So to confirm our observation, we inject SNE agonist intratumorally and see whether SNE agonist can induce monocytic MDSC increase like radiation treatment. So you can see, yeah, SNE agonist treatment can get us the comparable of increase of monocytic MDSC. So this suggests, yes, sting pathway can definitely recruit monocytic MDSC in tumor microenvironment after radiation. And this immunosuppressive factor may work as a negative feedback. So we want to know whether we get rid of this immunosuppressive population. So radiation will have a bad anti-tumor control. So to test this idea, we first utilized CCR2 knockout mice. CCR2 is specifically expressed on monocytic MDSC, and 95% of CCR2 expressed on monocytic MDSC. If we knock out these mice, so monocytic MDSC will not traffic to tumor sites from bone marrow. So you can see radiation, this radiation alone, if we inject tumor cell into CCR2 mice, radiation will effectively control tumor growth, and most of the tumor disappeared. So this suggests, in the absence of CCR2, and the tumor will get well controlled after radiation. So this genetic depletion of CCR2. So next we want to whether we can utilize the neutralizing antibody against the CCR2, and whether it can synergize with the radiation. So you can see, this combination therapy compared to radiation alone. The combination therapy can have a better tumor control, especially when we combine it with radiation, CCR2 neutralizing antibody, and SNE agonist. They have the best tumor control. So this suggests after radiation, there's an accumulation of monocytic MDSC. The reason is that CGAS pathway can induce interferon production. This production will induce the generation of CCR2 7 and 12, which is the ligand of CCR2, and this will this mechanism will cause the accumulation of monocytic MDSC. And if we get rid of this subset with CCR2 ablation, genital ablation, or blockade antibody ablation, so we can have an increased CD8 T cell response and have a better tumor control after radiation. Next, I want to share with you our unpublished data. As we all know, cytosolic DNA is a danger signal, and we first we test where the DNA comes from. So as we all know, in tumor cells, DNA may come from nuclei or from mitochondria. In our system, we find radiation can induce genomic DNA in a cytosolic, and also mitochondrial DNA can also is enriched in the cytosol of tumor cells after radiation. And next, we want to know which DNA is a major contributor for interferon beta induction after radiation. To test this idea, we utilized a protocol. In this protocol, we utilize HBr and to get rid of mitochondrial DNA. And so you can see if we deplete mitochondrial DNA, so the interferon beta is significantly decreased. So this suggests that mitochondrial DNA is a major contributor for interferon beta production after radiation. Similarly, we also find SNE activity is decreased after mitochondrial DNA depletion. As you know, the current dogma is that micronuclei is a region to provide DNA to trigger CGAS pathway. So they think the genomic DNA is a major contributor to induce interferon after radiation. So our observation is inconsistent with the current dogma. So next, we want to know what's the mechanism. Why mitochondrial DNA is released after radiation?
Excuse me, Dr. Deng. Do you have a lot of slide?
Okay, just one slide.
Okay.
So when we so very interesting, we find if MLK and ZP1 pathway contribute to type interferon production. If we look at interferon beta production, also you can see a SNE activity is decreased after the depletion of MKL and ZBP1, because MKL and ZBP1 contribute to mitochondrial DNA release. This the reason, or this mechanism what we found. Here is our model. We think releasing this ZBP1 and MKL pathway will induce mitochondrial DNA release, which will cause CGAS pathway and induce interferon beta products and activate the innate and adaptive immune response and contribute to anti-tumor effects of radiation. So yeah, here's the end. So I stop here and would like to have your questions.
Thank you very much, Dr. Deng. Thank you for this very nice talk. And is there any question from the audience? No? I have a question, Dr. Deng. Actually, you showed that STING is able to be phosphorylated. Do we have any ideas about the kinase and involved in this phosphorylation? And is there any different kinases able to phosphorylate STING?
That's good questions. So I think there's multiple. I think this multiple proteins contribute to the STING phosphorylation. First, I think the first one is the CGAM, which is the second messenger to induce STING activation. So in our finding, we find ZBP1 and MKL pathway also contribute to STING activity. But we can see, yeah, this contribution is not direct; it's indirect. So we have another study. I didn't talk about it here. We find metabolism can contribute to STING phosphorylation, like mTOR2. But we still figure out what's the mechanism.
Okay, because actually, when you treat the cells with ethidium bromide, it looks like you change the level of phosphorylation of STING. So right, I was wondering if triggering the kinase won't be a good way to also to regulate or to control the activity of STING.
Yeah. I think, yeah, just like when we utilize the STING agonist alone to treat a tumor, but it doesn't work well in vivo. So we think STING signaling alone activation cannot induce a robust anti-tumor immune response. You know, okay, radiation can generate multiple effects. It not only induces the STING activation, but also induce antigen release. The two events work together to generate robust immune response.
Okay. And any other question? Okay, so maybe we are going to move on because we are a little bit late. So thank you very much, Dr. Deng, again.
Thank you, everyone.